This is a production of Cornell University.
Thank you, Mike. Thank you, everyone, for being here today. So we all try to maintain a healthy and diverse diet, but this is not always easy. Although nutrient deficiencies are far less prevalent in the developed nations, surprisingly large proportions of the US population still do not obtain the daily recommended amount of several nutrients, including iron, zinc, vitamin E, and certain carotenoids. What if I tell you that we could help with that by increasing the nutritional quality of sweet corn? And that's what my project is about. So carotenoids are compounds produced by plants, and some of them have provitamin A, while tocochromonols have vitamin E activities. As antioxidants, they might decrease the risk of several health complications, including cardiovascular disease, age-related macular degeneration, which is the leading cause of blindness in people over the age of 60, and specific cancers. Certain studies have shown that 26 to 37% of the US population have low amount of vitamin E, and this might be related to increased risk of cardiovascular disease. Also, age-related macular degeneration affects 6.9 of adults of the age 43 years older, and it could be up to 25% if we consider older than 65. And in general, children and the elderly are the most vulnerable group for nutrient deficiencies. Nutrient deficiency is rare in the US, but inadequacies are common. This is when you don't have symptoms, but you are consuming less than the recommended amount. And this could lead to long-term health complications. This happens because 75 to 80% of people do not consume the recommended amount of fruits and vegetables. And I can see a lot of fruits in the back still, so. And also, the horticulture products has decreased the amount of several nutrients in the past few decades, particularly for iron, copper, potassium, and calcium. As you can see here, for some of those elements, there are a huge percentage of people that do not consume the recommended amount, particularly for magnesium, calcium, and zinc and iron. It is estimated that 9% of children and 14% of women have low serum ferritin, which is how iron is measured. And this and they might be at risk of having anemia. So biofortification could help with that, and sweet corn is a good option. US is a major producer of sweet corn, and sweet corn is the third most consumed vegetable in the US. Also, some studies have shown great variability for tocochromonols and carotenoids in sweet corn. As an example, this study in Australia have shown they were able to significantly change the carotenoid profile in sweet corn, and as you can see, they were able to change the color from yellow to more orange. And that could be also a good marketing tool to distinguish the product. Carotenoid and tocochromonol pathways are well known, and several genes have been identified and to be associated with natural variation in maize. Here you can see several compounds for carotenoids and tocochromonols. So for now on, I'm using the colors orange for carotenoids, blue for tocotrienols, and green for tocopherols. And from these pathways, the main ideas to remember is the alpha, beta carotene, and beta cryptoxanthin are provitamin A. Lutein and zeaxanthin were associated with decreased risk of age-related macular degeneration. Alpha tocopherol has the highest vitamin E activity, and tocotrienols in general have higher antioxidants. So the overall goal of my project was to dissect the genetic basis of vitamin and mineral levels in fresh sweet corn. And in order to achieve that, we try to identify causative genes and favorable alleles responsible for natural variation and develop and validate marker-based prediction models. To achieve that, we used a diversity panel with about 400 inbred lines from public breeding programs across the US. The inbred lines have mutations in genes from the starch pathway, which is particular of sweet corn. The two main ones are sugary one, which is a mutation in this enzyme on the pathway, and those lines accumulate more phytoglycogen, which is responsible for the creamy texture, and about 5 to 10% sugar. So that's the normal sweet corn. And then there is the shrunken 2, which is also called super sweet because it can accumulate 4 to 10 times more sugar, and particularly sucrose. And it's a mutation in this gene in the pathway. Visually, they look quite different at maturity. So you can see the sugary one is more a glassy type, while the shrunken 2, it's way smaller, the grain. But this is the maturity. So we were analyzing at fresh stage, which is when people consume it, and they look pretty much the same. So you cannot tell them apart just by looking at them. We also genotype using GPS, and we generated about 170,000 SNP markers. Using that information, we constructed a PCA, and as you can see, there's just weak patterns of population structure. And this is defined mostly by the star-deficient endosperm mutations. So there is a small separation from sugary 2 and shrunken 2 and sugary 1. But it's still the variation explained for PC1 is just 5%. The panel was harvested at fresh stage, which is about 21 days after pollination. And since it was fresh, we flash frozen using liquid nitrogen, we shelled and we ground the samples. For the carotenoids and tocochromonols, they were analyzed in Michigan State, and we use HPLC. We also analyze the minerals, but for that we use a different approach, and I'm going to talk about this later. Using models to estimate the BLUPs, the best linear unbiased predictors of the lines, you can see that most of the variation is due to genotype. There are a little bit of ear and genotype by environment interaction for some of the traits, but the majority is genotype. And because of that, the heritability are pretty high. As you can see, most of the traits have heritability of 0.8 or 0.9. So that's good for us breeders because it means that we can make more improvement by breeding those traits. Correlations also are mostly positive between the compounds. That means that we can improve one compound and still increase the other ones together. And this is true even for compounds from different classes. As you can see here, carotenoids and tocotrienols, they have moderate correlations. And that's probably because these compounds they all accumulate in the endosperm. Tocopherols, however, accumulate mostly in the embryo. So you can see there's not much correlation to the tocotrienols, for example. So that means that we can target both together and increase both traits. As for health benefits, one million fresh year of sweet corn can provide about 4.5% of the RDA for vitamin E and 3.5 for vitamin A. This doesn't seem a lot, but several studies in the past have shown that we can significantly increase some of those compounds. Also, we observed that the favorable alleles from the genes that increase those compounds, they are not all in one line. So we are still able to cross the lines and make and put all the favorable alleles together. So we still have room for improvement. For the sweet corn, the main thing is that it's a good source of zeaxanthin, which is one of the compounds related to the decreased risk of age-related macular degeneration. However, there's no official recommendation. If we use the levels that was used in the studies that found the associations, the supplemental levels, which is 2, you can see the sweet corn with just 100 grams, it can provide already 50% of that. And that's just to start with. If we compare to the paper, to the study from Australia that I mentioned before, we have here in our panel maximum 1.07. They started with half of that, with 0.048. After two generations of selections, just for increased total carotenoids, they were able to get 1.1, which is about what we already have. And later, they did more selection to target just one branch of the pathway, and they were able to get 3.07. So that means that with just like 100 grams, which is about 1 year of sweet corn, you can already achieve more than the supplemental dose. They also observe an increase in beta carotene, which is really promising because that's the main provitamin A compound. Even though they did not select directly for that, by doing these selections, they were able to go from 0.2 to 0.93. So that's a significant increase. And our panel has about the same as what they have. So that's good for us to start with. So we developed we conducted a genome-wide association study to see what genes are responsible for the natural variation. Here you can see the Manhattan plot for the ratio of these two compounds, delta to gamma tocotrienol, which are here in the pathway. We found association close to this gene, VT1, which is located here right before each of the compounds. We also analyzed alpha tocopherol, which has the highest vitamin E activity. And we have one significant marker within the gene that is right before alpha tocopherol. So it's responsible for converting gamma to alpha tocopherol. Something particular of this panel is that we found association of the starch genes with tocotrienols, which accumulate in the endosperm. As you can see here, the two greatest peak are close to sugary 1 and shrunken 2 that I mentioned before. And we also found association with AGGT1, which is responsible for producing precursors for all tocotrienols. So it makes sense to have association with total tocotrienols. When we classified the lines according to their endosperm mutation type, we noticed that for tocotrienols, the shrunken 2 lines in general, they have higher delta and gamma tocotrienol. One of the reasons could be because most of the lines are fixed for the favorable allele of the AGGT1 gene. So that means that they already have the favorable allele in that gene, for that gene, and they are producing more precursors. However, even if we compare just within the same allele, we can see the shrunken 2 lines is still accumulate more tocotrienols than sugary 1 lines. So that indicates that there might have something else going on there. However, we could not test that because of the confounding effect that most of them are fixed. So in order to study that, we are planning to use isolines. So those are lines that have the same background, they just are different in the gene of the start gene mutation. So we can compare the accumulation of those compounds and also RNA expression. And doing that, we expect to see if they do have any direct association with the levels of tocotrienols. In the literature, however, we have found some studies in our Arabidopsis that by increasing sucrose, they were able to increase the activity of the enzymes in the MAF pathway. So by increasing this pathway, they were able to produce more precursors. So our hypothesis is that by having more precursors, these lines would produce more tocotrienols. But as I said, this is something we still need to test. Although we don't have a good explanation for the sugary one lines, we think that those associations with that region is just because of the high LD, the linkage disequilibrium between the two genes. Because most of the lines have one endosperm mutation or the other. So that's why we are seeing those associations. So white lines, they have low carotenoid content in the endosperm. So if we do, for example, use a phenotype, just a binary phenotype, white versus yellow, we will find a large effect close to this gene, PSY1. And this is in this part of the pathway. So that means that depending on the allele for that gene, the line doesn't produce any of the carotenoids there. So in order to study the quantitative variation, we excluded the white lines for the carotenoid analysis. So for the genome-wide association for carotenoids, we found for beta carotene, which has the highest provitamin A activity, we found one association with QRB1, which is responsible for degrading beta carotene to transform into beta cryptoxanthine and further to zeaxanthine. For the ratio of beta xanthophiles and beta to alpha xanthophiles, which are located here in the pathway. So this is the alpha branch and this is the beta branch. We found one large association with LIKE1, which is here in the pathway, and it's responsible for changing the flux to one branch to the other. So depending on the allele, you're gonna have more alpha xanthophiles or beta. And in our panel, we actually noticed that most of the lines have the favorable allele for the alpha branch. So we have room to select for the lines that favor this branch, which has beta carotene and zeaxanthine. Other than GWAS, we tested genome prediction models as an approach for genetic improvement of sweet corn. Using GPLUP with about 170,000 SNP markers, we were able to achieve prediction accuracies about 0.5 to 0.7 for carotenoids, 0.4 to tocopherols, and 0.65 for tocotrienols. Because we saw association with the genes in the starch pathway, we tested including that information in the models to see if there is any increase in the accuracies, in the predictive abilities. As you can see here for tocopherols and alpha tocotrienol, by including the kernel type in the model, there's no difference. However, for delta and gamma tocotrienol, we saw about 5% increase in the predictive abilities. So that means that this information is not being captured by whole genome-wide SNP markers. And these are the same two traits that I showed before, that there was a difference between the two classes of lines. So we use it. We use the genome prediction models for the main carotenoids and tocochromanals to select the best 20 or 25 lines, and we separate it into two classes of endosperm mutation. And we used the genes that we identified in GWAS, and we score each of those lines to see if they had the favorable or the least favorable allele in that gene. So we had the 3 genes for tocochromanals and 2 for carotenoids. And here in this table, the light color, it means it has the least favorable allele, and dark color is most favorable. As you can see, there's no row with all the best combination for those genes. So that's our job as a breeder. We then cross lines with complementary traits, complementary alleles. For example, I would cross this one to these 2 rows. So in the end, we expect to select lines with both favorable alleles in those 2 genes. So far, we have made over 150 crosses, and they were self, and we are going to start validating the models in the near future. As a take home of this section of the presentation, we have identified genes in the pathways for tocochromanal and carotenoid pathways, especially like 1, CRB1, VT1 and 4, and HGGT1. We also saw some associations of sugary 1 and trunking 2 with tocotrienols, and that's the thing that is the most different thing from my project that was not reported before. We also showed that sweet corn has a good contribution of zeaxanthin, which is important for decreasing age-related macular degeneration, the risk of that. Predictive abilities were moderate, so that means that we can use those models to select the best lines. And the first crosses were already made to increase tocochromanals and carotenoids. For the second part, we study the ionomics, which means the mineral content of those lines, because that is more is associated more with environment, and then there's a high genotype by environmental interaction. We used the same samples, but we had an extra location. So we measured here in New York and also in Wisconsin. The lines, the kernels were lyophilized, and then we used ICP-MS to measure individual seeds, individual kernels. As you can see here, there's a similar plot that I show for tocochromanals. Genotype still has a great variability, is responsible for most of the variability, but now you can see a lot of environment playing a role and some environment, genotype by environmental interaction. Still, the heritabilities were moderate. So for iron and zinc, for example, is 0.63 and 66. So it's still good enough for increasing by breeding. As correlations, they are mostly positive between the nutrients as well, particularly for the ones that have similar chemical properties, such as calcium and strontium here. And magnesium and phosphorus were high too, and as well as iron and zinc. And that's probably related to the transport and accumulation in the plant, because several transporters of iron, for example, they are the same for zinc. One thing that, one down point of this is that phosphorus was positive correlated to zinc. And this is not ideal because phosphorus is mainly accumulates as phytic acid in the kernels. And phytic acid is known to decrease the bioavailability of iron and zinc. So that means if you are eating a grain with lots of iron, but it has also phytic acid, then you're not going to be able to absorb as much. When we compare the values between locations, we can see that there are differences between the locations, but there's no trend in favor of one location or the other for most of the nutrients. So some nutrients were higher in one location and the others were higher in the other. Iron and zinc, there was no difference. And iron, even though there was a small difference, you can still see variability in both places. For the genome-wide association study for these traits, we found significant associations for zinc and iron in the same region. And this is close to the nicotinamide synthase gene, NAS5. So NAS is here, and it's responsible for producing nicotinamide. So this has essentially two functions. It serves as a precursor of mugenic acid. So mugenic acid is used for grasses, gramineous plants when they are deficient to iron or zinc. They secrete these molecules in the soil, and these molecules form complexes with iron and zinc, and this helps them to uptake in the roots. Another function is that they serve as they are important metal chelators. So they form the nicotinamide form complexes within the plant and are responsible for transporting. And there are basically two types of enzymes, the NAS. So there is class 1, which produces the nicotinamide, which is used for mugenic acid or for long distance transportation. And there's class 2, which is more related to homeostasis and iron detoxification. If we see the enzyme that we found, the gene that we found association is NAS5, and this is that class 2 enzyme. So that indicates that the variation that we are seeing are mostly due to the redistribution of iron within the plant. So basically going from leaves, for example, to the kernel. Another association we found was with cadmium, and this is close to a heavy metal TPase. If we see here in the root cell, cadmium, after being entering the cell, it forms complexes with phytokeratins, which is here, and these complexes are then sequestered by HMA3, which is the association we found. So that means that some plants are able to sequester the cadmium in the cell roots, and then that cadmium is not transported to the kernels. One thing to note is that although we saw a variation for cadmium in sweet corn, even the highest accumulated one, it still accumulates just really small amounts. If we consider the toxic amounts of cadmium for people, the line that accumulates the most, it would provide you just 5% of the maximum that you could have with 1 year. So it's still really far to be worried for now. But of course, if we increase iron and zinc, we should still keep track of cadmium to make sure that's not an issue. We also analyze the concentrations within each location to see if there's any specific association. For molybdenum, we found one association in chromosome 1, which we don't have any candidate gene so far. There is this known transported of molybdenum in the same chromosome, but it's quite far. So you can see that we had some signal there, but we didn't have enough power to detect. We also found association with nickel in New York, and this is close to PTR2, which is a peptide transported. So peptide transporter, they transport mostly nitrate, malate, and histidine. And it's also known that nickel binds with histidine with high affinity. And there was some association of histidine in the detoxification of nickel in plants that accumulate a lot of nickel. So although we did not find a direct association between this transporter with nickel, we hypothesized that this transporter might be transporting a complex of nickel plus histidine. But this is just this is what we are thinking that could happen. We still need to test that. And for Wisconsin, we had one specific association close to this gene, RAS2. So RAS, like GTPases, are regulated by calcium-dependent mechanism. And some small GTPases were shown to influence calcium channels as well. So that's likely a candidate. But also another thing that could be happening is calcium is highly correlated with strontium, which is another metal with similar chemical properties. And in the nascent association mapping of maize, they found one significant association in the same region, pretty close to our marker, but that was for strontium. So we're still not sure if this association is particular with calcium or something related to. We also use genome selection for these elements. And as you can see, they were a little bit lower than carotenoids and tocochromanos, but they vary from low to moderate. And the two main nutrients that we are interested in, which is zinc and iron, they had above average predictive abilities. And they were mostly associated with heritabilities. So the more heritable traits, they had more predictive abilities. We also tested the kernel type to see if we could increase those predictive abilities. And in this case, it did not change the predictive abilities for any of those nutrients, as you can see here. And one reason for that is that differently from carotenoids and tocotrienols, which accumulate in the endosperm, most of these elements accumulate in the scutellum of the embryo, which is this part you are seeing the colors here. So for most of the elements, you don't see much in the endosperm. There is a little bit for potassium, but it's still, you can see that it's blue, so it's low compared to the scutellum here. As a take home of this part, we identify several genes that are associated with iron and zinc, NAS5, cadmium, HMA3, and two associations specific to nickel in New York and calcium in Wisconsin. We also noticed that sugary 1 and shrunken 2, they didn't have a big effect on those nutrients. It was not as pronounced as carotenoids and tocochromanos. And iron and zinc, they do not seem to have the same genetic control as cadmium. So that means that we could increase those two elements without increasing cadmium. So that's good for us. And we saw moderate predictive abilities, particularly for iron and zinc. So this is just the first step. So far, we have used the prediction accuracies in our results to select lines for carotenoids and tocochromanos. So we were able to start the breeding program. But in the future, we expect to use the rest of the results to increase even more other nutrients as well. So we want to have corn, like fresh sweet corn with more nutrients in general. I would like to thank the committee members, my committee members, my advisor, Michael Gore, Margaret Smith, and Gavin Sachs. Also the whole Gore lab, which helped a lot with the field trials. So we all went to the field and helped planting and harvesting. So particularly Nick Hasmar, which was always responsible for in charge of the field activities, and Jenna for taking care of the project from now on. Also, all our collaborators from different universities, my funding, Cornell and Capis, and all my family and friends. And I think my mom is watching on the internet. Thank you for your attention, and I will be happy to answer any questions now.
We have ample time for questions.
So on the applied breeding side, Matt, maybe just the pictures you showed, but they were slightly sad looking. Here's the sweet corn. I know that it's been kind of difficult growing conditions the last few years for corn around here. So do you think it's just environment, or are these more unadapted genotypes, or do you think that, I guess the question is, can you get all these carotenoid and subretinal copperol genes into more elite germplasm for sweet corn easily? Because for fresh market sweet corn, like the appearance of the cob is pretty important.
Yeah. So the question was about the appearance, if we could transfer those nutrients to a more appealing sweet corn lines. So the thing is that those are inbred lines, and they usually look really bad in the field. They are really small, and the pictures that I show, they are all hand pollinated, so they were manually done. So there's a lot of variability depending on the day, depending on who did it, and the year. So for the open pollinated ones, they look pretty good. And if we plant those, like those would be hybrids, and we expect to have better quality. So that's the main reason.
They also taste good for the ones I was able to
Is there any upper limit that you have to worry about with vitamin A and vitamin E in the future about having too much?
So the question was if there is an upper limit that we should take care to not go beyond it. So for vitamin A, probably not, because the plants, they don't produce vitamin A, it produces carotenoids, which are pro-vitamin A. So it's converted to vitamin A in the human body. So if you eat too much carotenoid, your body will just not convert to vitamin A. So that would not be an issue. And for vitamin E, by looking at the numbers that we have, I don't think that would be an issue also, because so far it's still quite low, like 4.5. So I would think that would be able to increase like to 15%, like 20. I don't think it would go, and then 100% is what you need. So you still have a lot to go over to increase, like to go to upper limit. So I don't think that would be an issue. Yeah. Yeah, most of the toxicity of those vitamins is when people take supplements. They usually not come from consumption.
You had the one association with the ratio between the two branches, and I guess my question first is, did you test GWAS for that ratio because you knew what the biochemical pathway was, and therefore it was sort of a priori motivated?
Mm-hmm.
And if that is the case, how would you go about sort of finding those types of associations if you don't know, if you're working on some metabolite that you don't know the biochemical pathway? I mean, or is that just impossible?
Yeah, so the question was why we use the ratio and what we had in mind, and what if we didn't know that before, what we would have done. So yes, we did use because we knew the pathway, and because previous work have used the same ratios. So if we didn't know the pathway, I think I would first look at what other people did in the area and look for correlations to see if they tend to accumulate together or see something about the biology. For example, for the nutrients, the elements, they are not part of a pathway. They are all separate, but they are correlated, like they are a whole network. So what we did test just two ratios of iron and zinc divided by phosphorus. And that's because we know that phosphorus could be decreasing the bioavailability of iron and zinc. So what we had in mind was trying to test the best combination that like you had more iron but less phosphorus. So that's what we try to do for the ratio. So it depends the what you want to test.
Here's a point of order question to Michael. Can I ask questions now, or is this a saver for the? I'll cut it down the middle of the so hopefully it won't take too long. You are interested in accumulating high levels of iron and some other trace nutrients, but your concern is bioavailability, and you're using phosphorus and phosphoric acid. Has that been validated? That is an appropriate proxy for bioavailability? And then more generally, if you had a way to alter the chemical composition, what would you choose if you wanted to achieve high loadings of iron and ensure that it was bioavailable? What's the right way to accumulate more iron in corn? What's key?
Okay, so I wish I had a good answer for that, but
I'll save it for this afternoon.
yeah. So the thing is, it was validated that it is a proxy, but it's not perfect. Like it is, we know that 90% of the phosphorus is in the form of phytic acid in those kernels. And we know that there is a true association with phytic acid with bioavailability. It's not one-to-one, but there is association. So the best way to test that is using KCO2 models, for example, where they actually test the bioavailability. But in our case, since we didn't have that, the best way was to use the phosphorus as a proxy. And concerning about what I would do, I think I would try to decrease the amount of phytic acid, but just it needs to be done just in the seed. So there has been some studies in the past that they did that. So you cannot decrease phosphorus in general because maybe that phosphorus is needed for the germination of the seed later. So some of studies, they decrease the amount of phytic acid. So they change some enzymes so the phosphorus keeps as phosphorus or inorganic phosphorus instead of phytic acid. And that's best for the bioavailability. But also that needs to be done just in the seed because phytic acid is good, like it's usually the whole plant. So if we change the whole plant, there are other things that might change too. So the quick answer would be like trying to decrease phytic acid, especially in the seed, but still keeping similar levels of phosphorus. It's in the same, unfortunately, it's in the same region of iron and zinc. So it's in the embryo.
So what about cooking? What do you know about how any of this holds up through cooking? I love sweet corn broth, but not everyone does.
Yeah, so the question was about cooking, how the nutrients are after cooking. So actually carotenoids, they are more available after cooking. So there has been some studies that after cooking, they are more, you are able to use more than before cooking. And for the nutrients, there has been some studies that the phytic acid is broke down also. So in theory, iron and zinc would be more bioavailable because they are still there, but then phytic acid is breaking down, and then they are not forming complexes together. So in general, keep similar or be better, actually.
You showed some genomic prediction where you added the kernel type in as a predictor, and it looked like you were just adding them in as additive predictors. You know, I mean, I guess particularly for the sort of kernel, is that right? If there might be an interaction between the kernel type and how those accumulate in the kernel. So in other words, some kind of epistatic interaction between the kernel type and the rest of the genome for accumulation of.
Yeah, so that's actually what we don't know, and so far we cannot test with the panel that we have because of the confounded effect, because most of the lines, they have the favorable alleles for those genes, so we cannot compare with one or the other. That's why we are planning, we have like 2 or 3 backgrounds, like isolines with different backgrounds, and then we can test exactly that to see if there is any interaction between those genes and genes from those pathways. So far we don't have an answer for that.
Could you grade blue sweet corn, like starting to put other pigments in sweet corn?
Other pigments?
Yeah, like blue and.
So there is Bill Tracy, which is from Wisconsin, and he has been doing that actually. He was able to have he has a new line now. It has more anthocyanins, which accumulates more like a purplish. It's still, he has a new line and it does look pretty nice. It's like, it's not the whole year, it's just like a couple of kernels. But there is some, the hard part is that anthocyanins, they are accumulated later in the maturity. So when it's mature, it looks pretty good. But then when it's fresh, it still has some color, but it's not as dark. And the other thing that he noticed is that after you cook, all the color leaches out. So it's nice if you are like buying, but in the end, it's more like appealing for buying. You don't, when you are eating, you're not gonna see a lot for now, but that he might be able to improve that. Yeah, it is good. Like I did try raw and it was okay. It was good.
So at least for the vitamin part of this, and I don't know about the metal part, but most of these pathways are pretty well described. And so I guess my question is, what was the justification for doing genome-wide scans as opposed to just looking in region, right? So you could have said, okay, we know that these 25 genes are in this pathway. Mm-hmm. Let's tag those regions with the SNPs that we have in right in that spot. You just test those SNPs. And you know, additionally, by doing these genome-wide scans, did you detect anything that you wouldn't have detected by just using known pathways?
So the question was about the reason why we tested the same. We didn't test only like the candidate genes. Right. So I have 2 good answers for that. One is that we would not have found the start genes because we would never think about those as candidates. And the other thing is we did test genome prediction with SNPs only close to the candidate genes. And the predictive abilities were quite low compared to using genome-wide. So for tocotrienols, it would drop like 10% or less. So we still think maybe we don't have enough coverage in those regions, but then, and yeah, it's basically that. I think we don't have enough coverage, and maybe other genes are also have a part of that. So the main reason would be finding like new associations.
Kind of as an actually unrelated follow-up. For the starch genes, is the association that you found there just associated with the fact that they have less starch at higher concentrations? Or
we thought about that, and we did include kernel weight and like moisture, and it did not seem to explain. Okay. Because there's a high difference in weight, but it's at maturity. So at fresh kernel, the weights are pretty much the same. So there is a difference. It's about 1 gram, like 1 milligram difference. But when you use in the models, it did not explain.
You mentioned that the prediction ability is low if you just use sleeps from the candidate genes. But in your slides, the major side you showed nearly all from the pathways. Why you did not identify maybe any transcript factors that may affect multiple pathway or affect specific pathways in your studies?
The question was why I did not find associations with other transcription factors that affect the. Is that
beyond pathway?
So I think the main answer would be probably we don't have enough variability in the panel because most of the lines are derived for a few lines in the past, or we just didn't have a power to detect, like maybe it's like a smaller effect and we didn't have a power to detect those.
So the number of transcription factors we found controlling brought no variation in base rate and base and population was only 1. Yeah, and that explained maybe 1 to 2% of the total phenotypic variance. So I'm not surprised we didn't find it.
Yeah, and then the NAM population has 5,000, we had 400. So I did not expect to have power to detect them.
All right, we'll let Matt recover from this so you can have this PE exam at 3 p.m. today, but let's take him first.
This has been a production of Cornell University, on the web at cornell.edu.